Hello everyone, welcome to this InSphero webinar. We are going to wait another minute so that everyone can join online. In the meantime, you have on the right side some polls where you can answer a couple of questions related to this webinar. There is also a chat window that you can use to ask questions during the whole duration of the webinar, and we will answer them at the end of our presentation. Hello everyone, welcome to this InSphero webinar called Predicting Metabolism Related Drug-Drug Interaction with Organ-on-Chip Technology. I'm Olivier Frey, Head of Technologies and Platforms at InSphero, and I'm happy to be with Christian Lohasz, which is a postdoctoral researcher at the bioengineering laboratory of the BSSC at ETH Zurich.
Hello, everybody.
So the presentation that we will give is the fruit of one of the collaborative efforts that InSphero had with ETH in the past, where we developed a microfluidic system, which will be the first part of the presentation that I will give. And we use this microfluidic system, Akura flow organ-on-chip technology, for a specific case study looking at drug-drug interaction. And this will be the part of Christian. So organ-on-chip technology is a technology that has advanced quite significantly in the past few years. Basically, two major tracks in the development. So the first one, the first element is the evolution of cell cultures. They have become more and more complex, from 2D cell cultures with single cells to three-dimensional tissue structures that incorporate not just a single cell type, but multiple cell type to closely replicate a tissue structure in vivo. In parallel and initially disconnected, we had the evolution of devices based on microsystems technology. Microsystems technology has been used in IC industry and afterwards in so-called MEMS, microelectromechanical systems, that are now sensors and actuators used in every cell phone. The same technological processes have later been used also in microfluidics. So small microchannels have been structures and substrate so that miniature and small amounts of liquid can be handled, which are at the same scale as you have it in the cells. These microfluidic systems then in a later stage have been used to actually culture 3D tissue culture. And here the two technologies merged, enabling the construction of so-called organ-on-chips. So where at the beginning we had 3D tissue cultures that were mainly used on the same single dishes, now the 3D cell culture systems can be incorporated into specifically designed cell culturing system fabricated by microsystems technology. So one of the examples can be seen here on the right side from the Fuji lab, where we have cells, three-dimensional cells that are incorporated inside a small channel where they are building three-dimensional structures, and the device has been engineered in a way that they incorporated endothelial-like barrier using very small channels that mimic this endothelial barrier in vivo. So this is a very nice and very early example of such kind of a liver on a chip. A next level of complexity in engineering in vitro systems are so-called body-on-a-chip systems or multi-tissue systems that allow to study organ-organ interaction and emulate systemic effects that today are only available in vivo, in animals or human. So this is a very a schematic view of such kind of a multi-tissue system where we have a couple of compartments where the organ models are located in. You have microfluidic channels that interconnect these compartments, infusion ports to add compounds or substances, sampling ports to remove small amount of liquids so that you can analyze them, and a fluidic pump that lets the liquid flow and circulate between these tissue structures in a physiological way. So such kind of multi-tissue systems can be constructed in different ways. Let's assume that you have 3 different organ models: a gut barrier model, a liver sandwich model, and a pancreatic spheroid model. So they can be interconnected highly flexibly by just using tubing. And this is an example that you can see on the right side. It allows you quite flexibly to connect and disconnect these different organ models each other and create such kind of a multi-tissue system. A second version is a monolithic design, type number B, where the channels are structured directly inside the substrate and are hard-coded inside the device. This allows less flexibility, but at the same time, you can very precisely define the liquid routing between the different organ models using microsystems technology structuring processes. The last type, type C, is a mixture of both, where we have the fluidic channels and the compartment hard-coded inside the device, as for example, the fluidic layer, as the company Tissues is doing it here in the middle. And then you have plug-in models that are inserted into device from the top into this microfluidic channel structure. So these different devices have advantages and disadvantages. An important point when going into more systemic multi-tissue systems is the yields and the reproducibility of the whole system. And this is highly depending on the yield and reproducibility of the single elements, which is nicely illustrated here. So if you have a flexible system where you first produce the different organ models and you do a quality control and connect them afterwards, as for example, the tube connected system, your yield is very high and is independent on the number of organs that you integrate into such a system. However, if you have a monolithic design where all the organ models need to function during your multi-system experiment, your yield is decreasing with the number of organs that you're integrating into the system. Because you can imagine if just one of these models, for example, the gut, is not working, your whole system will collapse. So what we used at InSphero and ETH is type C. So what we're doing is we rely our microphysiological system on our core technology, the microtissues. We use industry-validated system to produce these microtissues in 384 and 96 well plates and QC them after the production. This gives us certainty that these spheroid models are working. At the same time, the same manufacturing process can be used for a lot of different models, as for example, the liver, the pancreatic islets, or tumor models. Once QC'd, we transfer them into our microfluidic system where they are combined in microfluidic channels to create a network and to expose them to more in vivo-like conditions, such as in vivo-like cell-to-liquid ratios, liquids turnover, and the dynamic mechanical forces that they would expect also in vivo. So the microfluidic system is something that I want to present you in more detail now. It consists in its core and a microfluidic chip with a slide format. And when you look at the cross-section, we see how the inside is structured. On each side, on the lateral side, we have two large reservoirs that hold 100 microliters of liquid volume. These two large reservoirs are interconnected by a straight channel. In this channel, we have 10 microtissue compartments that are depicted on the right side. One microtissue compartment is especially designed to hold a single microtissue so that it is preserved in morphology and does not merge. We have a liquid barrier, a liquid air barrier on the top that is kept open and restricts the volume within this small compartment. It's around 1 to 2 microliters in volume and therefore a very small dead volume. The compartment can be accessed from the bottom with every microscope. A very important part in this compartment is that we have direct access to the microtissue. So we can load the microtissue from the top into the compartment using this funnel-like structure where the tip is positioned, and we can remove the microtissue after an experiment for downstream analysis. These chips are then placed in a frame so that the single compartments are located in the 384 well positions so that it complies with SBS standards. We have 4 chips, which means that in the end we have 8 channels, which means that there's 8 independent experiments, and every channel has 10 of these compartments. For flow actuation in this microfluidic channel, we are placing these plates onto a tilter that is depicted on the right side. The tilter tilts the plates back and forth, for example, every 4 to 5 minutes, and the flow is generated by gravitational forces. One of the reservoirs is always at the higher location, and due to the hydrostatic pressure, we have the liquid that flows down the 10 microtissue compartments to the bottom of the reservoir and back when we tilt it back. So therefore, we don't need any fluidic connections, and we can stack multiple plates inside an incubator to run many experiments in parallel at a very small footprint. Also, we can remove the plates anytime for medium sampling, for medium exchange, or for imaging on an imaging system. An important point is that the complete system is fabricated out of polystyrene. And polystyrene has an advantage over, for example, PDMS with respect to compound adsorption and absorption. So we tested this characteristic by fabricating a polystyrene version and a PDMS version. We added different substances, 4 of them as depicted here, and we measured the concentration of these substances inside the channel over 2 days. And what you can see is the depletion, for example, of diazepam and testosterone on the very left side in the PDMS chip, because these 2 hydrophobic or lipophilic substances are very quickly taken up by the PDMS and not available in the channel anymore. Another characteristic is seen for acetaminophen and human albumin that are not absorbed into the PDMS, but at the same time we have most probably evaporation or just the water that is absorbed in the PDMS, and we have an up concentration of these 2 substances. On the very right side, you see profiles of a fluorescence dye, and you can nicely see how in the PDMS channel, you see, for example, Nile red that is diffusing into the PDMS, where we don't see any diffusion using the polystyrene material. So as I mentioned before, we need to transfer the microtissues from our production plates into the microfluidic chip to construct such kind of a multi-tissue experiment. And this is how it is done in a schematic view. So on the right side, you have the 2 production plates for example, liver microtissues and tumor microtissues, and we are transferring them directly into the Akura flow system on the left side by using pipette tips. So we pick up the microtissues from the production plate, we let it sediment to the bottom of the tip, and then we just need a contact of the pipette tip with the compartment so that the two liquids are merging and the microtissue is dropping into the compartment without any pumping actuation, minimizing cross-contamination from the production plate into the microfluidic chip. In this way, we can then transfer microtissues from different production plates, so different organ models, very flexibly into the Akura flow system. What you can see here is a picture of a real-world scenario done, example, using a 96 channel head. On the back, you can see the production plate, the Akura 96 well plate, and 20 tips that are located on the 96 channel head and transferring 20 microtissues at the same time. You see a zoom view of the tip with the microtissue, how it sediments to the bottom, and where it is then transferred on the left side into the Akura flow system. So with this direct transfer, we can transfer 20 microtissues at a time and load the full plate in less than 10 minutes. The whole system is also amendable for robotic transfer, where the whole loading can be done in a fully automated fashion. So a quick and mild transfer is important, especially if you think at going back to our initial example where we have different organ types that most probably have different fabrication and maturation times. So a gut barrier model most probably has a longer maturation time until it is ready to use. That's why you need to orchestrate the different fabrication processes, also if you purchase them from an external provider, in a way that at time zero, when you want to start and assemble your multi-tissue experiment, all these microtissues and all these organ models are ready, functional and viable, so that you can interconnect them and start with high reproducibility your multi-tissue system. And this is something which is important and needs to be considered. And having the microtissues in our system that have a long viability and functionality helps to very flexibly combine and construct multi-tissue experiments. So going back to my initial schematic view, what we did by engineering the Akura flow system is we use only microtissue spheroids, which is simplifying the system with means of organ models that we include, having all the same fabrication, a robust and reliable fabrication process. And with the Akura flow system, we linearize the system so that we can run multiple experiments in parallel at a very small footprint. And this was important for us because with this kind of engineering, we preserve the scalability that microtissues offer. A second advantage of this system of this approach is that we can now define a wide range of different preclinical applications by a very flexible arrangement of different organ models. We can just load liver into the system to increase the number of cells per liquid volume to address DMPK questions. We can, on the very right side, add the liver to target organs as the liver and the skin, for example, to look at metabolites that might be toxic for these organs. We can go into metabolic disease modeling, where we combine the pancreas and the liver, or even add the fats and look at the interaction of these organs, which are central in the metabolic syndrome. Or we can look at oncological questions, where we combine the tumor microtissues and the liver microtissues. And this is a case study that ETH conducted in the past years, and we will focus on in the next slide. So what they did is they combined tumor and liver, and they used this model to predict metabolism-related drug-drug interaction with this Akura flow technology. So this is now the time to hand over to Christian Lohasz, who will guide you through this case study.
Thank you very much, Olivier, for the very nice overview over organ-on-chip systems in general, and especially also for presenting your Akura flow system. Before I now get into the details of how we applied your chip system, I briefly want to take a little detour over the topic of pharmacokinetics. Pharmacokinetics is a term that generally describes what's happening to a drug upon entering the human body. In the first step, the drug is absorbed into the body, and the dynamics of this absorption process depends on the route of administration and on the nature of the compound. After being absorbed, the drug is then distributed throughout the body, mainly via the bloodstream. At this point, tissue-specific concentrations can build up due to local vascular permeability, blood perfusion rate, and binding proteins that might be present in one organ but not in the other. A very important step is then the metabolism of drugs. Metabolization mainly takes place in the liver, and where the compounds are modified and functional groups are conjugated. The resulting metabolites are then finally excreted from the body through feces, urine, or exhalation. The interplay of these four processes then defines the time during which the compound is present in the body and also the concentration at which the compounds are present. I now want to focus a bit more closely on the metabolism and especially in the context of prodrugs. Prodrugs are drugs that, upon entering the human body, need to undergo metabolism or metabolization before they can be efficient or effective. Here, our prodrug indicated in blue enters the body and is metabolized in the first step in the liver and more specifically by the cytochrome P450 isoenzyme family. The resulting metabolites, now indicated in orange, are then distributed again throughout the body before they can act on the target tissue, which in our proof of concept study was the tumor tissue. For our experiments, we chose two different prodrugs, cyclophosphamide and ifosfamide. Both of them belong to the same family of anti-cancer medication, and as you can see from the chemical structure, they are very similar. The small differences, however, make them to be metabolized by different CYP isoforms. While cyclophosphamide is mainly metabolized by the CYP isoform 2b6 and at a lower extent by CYP3A4, ifosfamide mainly relies on the bioactivation by the CYP isoform 3A4. Now, it has also been shown that the simultaneous administration of the antiretroviral drug ritonavir results in a loss of activity of the anti-cancer medication. This effect is mainly explained by the inhibitory function or activity by ritonavir on CYP3A4. This effect, where one drug affects the efficacy of another simultaneously given drug, is generally termed drug-drug interaction. And having this background information, we wanted to use the chip that Olivier just presented and replicate this behavior in an in vitro system. So in the first step, we wanted to characterize our metabolic component, which were the human liver microtissues also provided by InSphero. So we loaded human liver microtissues into the chip system and monitored their health and their functionality over a 2 weeks period. You can see the green data points that correspond to the liver microtissues under perfused conditions and the orange ones corresponding to the ones cultured under static conditions. On the top left, you see the viability dependent on intracellular ATP concentration over the 14-day period. Below that, you see the albumin secretion, which is a general marker for liver functionality. On the right side, you see the CYP activity of the CYP isoforms 3A4 and 2B6. And as you can see from all of these graphs, our liver microtissues cultured under perfused conditions behave very similar to the ones cultured under standard static culturing conditions. Now, it is very good that we have viable and functional microtissues in our chip system. However, we also need to know whether this functionality, especially the CYP activity, can be modulated by pharmacoactive compounds. This is why we also incubated liver microtissues on chip in the presence of the three compounds that I previously presented, ritonavir, cyclophosphamide, and E-phosphamide. Now on the top left graph, you see that upon incubation with ritonavir, the CYP activity of the CYP isoform 3F4 was inhibited, while in the presence of cyclophosphamide and E-phosphamide, the activity of CYP3A4 was induced. So here we could show that the functionality, the metabolic functionality of our liver microtissues was indeed dynamic and could be modulated. In the bottom right graph, you can also see that the viability was not impacted by the presence of the three compounds. In the next step, we then wanted to combine the metabolically active liver microtissues together with our target tissue, which was the tumor. We therefore loaded 6 liver microtissues and 4 tumor microtissues into the same microfluidic channel of the Akura flow system. We then exposed the microtissues to cyclophosphamide and E-phosphamide and monitored the growth of the tumor microtissues over time. This experiment was meant to show whether our microtissues, the two types of microtissues, could interact with each other, and whether we could see an effect of the prodrugs on the tumor microtissues. As negative controls, we also cultured 4 tumor microtissues on chip in the absence of liver microtissues. Now, looking at the growth of the tumor microtissues, we now see in the top panel the ones cultured together with liver microtissues. And from the growth curve, you can see that in the presence of cyclophosphamide or E-phosphamide, the growth of the tumor microtissues was inhibited. On the right side, you can also see representative images at day 0 and day 12 of the tumor microtissues. In the bottom panel, you can see the microtissues that were cultured in the absence of liver microtissues. And as you can see from the growth curves and also from the representative images, is that regardless of the presence of the two drugs, all tumor microtissues were able to grow over time. This experiment proved that cyclophosphamide and E-phosphamide require the bioactivation by liver microtissues. And after this bioactivation, the active metabolites show an effect on the tumor microtissue by reducing their growth. Having shown this successful interaction of the two microtissue types, we now, of course, wanted also to take a look at the drug-drug interactions. Therefore, again, we cultured the microtissues on chip with the two microtissue types, under exposure of cyclophosphamide and E-phosphamide. And here you see the growth curves that I already showed before. Now combining cyclophosphamide with ritonavir, we saw that the growth of the tumor microtissues was still inhibited to a certain extent. So the prodrug still showed an activity against the tumor microtissues. However, if you look now at the representative images, the tumor microtissue at day 12 appears to be slightly bigger than under the single drug treatment. Combining E-phosphamide with ritonavir, we saw that the anti-cancer activity of the prodrug was completely diminished, and the tumor microtissue could grow just as much as the control condition that did not experience any drug exposure. Statistical analysis of the 4 treatment conditions showed that we could indeed detect significant drug-drug interactions for both combination treatments when comparing them to the single drug treatment. Furthermore, the severity of the drug-drug interaction, which was much larger for E-phosphamide when compared to cyclophosphamide, was proportional to the involvement of CYP3A4 in the bioactivation of E-phosphamide. Having shown this effect on a morphological level by looking at the tumor microtissues, we also wanted to verify this effect on a metabolic level by taking a look at the metabolites that result from the bioactivation of cyclophosphamide and E-phosphamide. We therefore incubated our liver microtissues together with one of the two prodrugs in the presence and in the absence of ritonavir. In the activation pathway of the two compounds, the main metabolite with the anti-cancer activity is called phosphoramide mustard. However, This substance is very hard to detect by mass spectrometry due to its short half-life and the very rapid decay. We therefore chose a side pathway of the bioactivation pathway that produces the alco and carboxy metabolite of the two compounds. Being able to detect one of these metabolites also suggests that we have a functional bioactivation pathway by the liver microtissue, and it also suggests the generation of phosphoramide mustard. Looking at the metabolic footprint of cyclophosphamide over a 3-day period showed that the alkophosphamide and carboxyphosphamide could be generated in the presence and in the absence of ritonavir. So we did not have a full inhibition of the activation pathway using cyclophosphamide as a prodrug. Looking at the metabolic footprint now of ephosphamide, we could only detect alkoephosphamide, and we only saw the metabolite generated in the presence of the liver, but when also given ritonavir, the production of alkoephosphamide was reduced to an undetectable amount. So these data again proved or showed that we could detect drug-drug interactions in our chip system, which we already showed on a morphological level looking at the tumor microtissues. And at this point, I already want to hand back to Olivier, who will now sum up and conclude the talk. Thank you very much for your attention from my side.
So thank you very much, Christian, for this nice overview and presentation of this very specific case study where you use the Akura flow system. It's a great example how you can use microtissues that are produced, for example, in 96 well plates, as you can see here on the left side of the slide, and then simply transfer it into the Akura flow system, where you can have different microtissues that are interacting with each other, and where you can look at paracrine signaling, as for example, the metabolites produced by the liver microtissues, and then have an effect on the tumor microtissues. And it's exactly this platform that we now also want to make available to research groups all over the world with our Akura Flow Academic Access Program. So the idea here is to provide the system to research groups in academia that are used to work with spheroids or that want to get used to work with spheroids or microtissues, and quickly develop single or multi-tissue system using this powerful and easy to use plug-and-play microfluidic platform. So when you want to fast forward your research, please visit InSphero.com Academia, where you find all information and where you can apply for the Akura Flow program. With this, I want to share a couple of thoughts on the future perspective of our development. So on the technology side, we will move the current Akura flow system from the 8-channel version towards the 24-channel version, which includes that we transfer the plate format from the current chip version in a more 384 plate version that allows to have more channels in parallel and therefore more replicates on a single plate. In parallel, we are also working on including immune cells into the microfluidic channels. This requires some redesign so that the viability and the function of the immune cells are preserved when they are flowing through the channel. On the application side, we in parallel also will work on different applications because the Akura flow system allows to very flexibly combine different microtissues in the microchannel. And we will focus specifically also in the area of metabolic diseases because we have a very performant islet microtissue platform at hand, as well as the liver microtissue platform. In both platforms, we are able to drive the different healthy microtissues into the different disease states, making it very interesting to let them interact with each other. With this, I would like to end the presentation and thank all the contributors on the InSphero side the members in my team, the oncology team, the liver team, as well as the BPU. And on the ETH side, all collaborators that worked on that project, specifically also Professor Andreas Hierlemann, the head of the lab. Finally, also the CTI consortium, which provided the funding for this project. So the talk or this webinar is now open for questions. You also see the email addresses at the bottom of the slides for questions that can be addressed at the later stage.